So as I said, there are three classic signs associated with untreated diabetes mellitus, and those are polyphagia, or excess hunger, polyuria, or increased urine production, and I'll put it down here, polydipsia, which is increased thirst. So what I want you to do for me for just a second, hit pause, see if you can come up with an explanation for how or why each of these three occur. How does this all relate back to malfunction of insulin? So I'm hoping you really did hit pause and you really did try to figure out what these three P's mean. Part of the active learning process is really trying to see what you do and don't know. The more you interact with the material, the more likely you are to remember it. So let's go through each of these and see how close you were to figuring out what caused each of the three of these. And we'll start with polyphagia up top. So in polyphagia, if you think about this, when we look at the cells of the body, as I said before, they're starved for glucose. And so that's going to stimulate gluconeogenesis. As I said before, the body responds to the signal from cells that they're starved by trying to increase the amount of sugar available for cells to use. Of course, that doesn't work because you need to have insulin to gain permission to use the sugar that's being made. But we do get increased amounts of gluconeogenesis, and in order to fuel the production of new sugar, we need to break down intracellular stores. Intracellular, inside of cells, stores of molecules such as lipids, for example, as I said before. And the more we break those down, the more that's going to stimulate the hypothalamus to initiate the hunger sensation. So the person feels hungry and wants to eat more and more and more. Okay, so let's look at the second P, polyuria. Now you'll remember that in the kidneys, the filtration process that occurs in the kidneys initially allows for lots of different desirable solutes to make it into the forming urine, things like glucose and amino acids and different vitamins, etc. And then what happens in the renal tubules is reabsorption of all those desirable components that we want to keep, pulling them back out of the forming urine, in other words. And glucose is, of course, one of the main molecules that gets reabsorbed from the forming urine and put back into the blood. And that occurs thanks to the efforts of something called SGLTs, or sodium glucose transport proteins. And sodium glucose transport proteins, they have a transport maximum. There's only so many molecules of glucose per unit time that those sodium glucose transport proteins can absorb. Well, imagine, if you will, you have a person with hyperglycemia, an untreated diabetic. They have extremely high levels of sugar in the blood, which are then getting initially filtered out into the forming urine. And even though the sodium glucose transport proteins are reabsorbing sugar as quickly as they cannot absorb the excess amounts of sugar. There's just too much glucose present in the forming urine for the sodium glucose transport proteins to absorb it all. And so much of that glucose then is allowed to go into the forming urine, or I guess I should say remain in the forming urine. And glucose has, because I have more glucose in the urine itself has a higher osmolality. Right? It has more particles per unit volume. And as a result, it tends to draw water to it. So more water stays in the urine or gets drawn into the urine. And as a result, the volume of urine is higher than you would see in a normal person. What about polydipsia? Polydipsia, you'll remember, is a sensation of increased thirst. Well, this should make sense. If I'm losing lots of water in the urine because the urine has such a high osmolality due to all this glucose present, well, that's going to stimulate the thirst centers in the hypothalamus, and that's going to stimulate the desire to drink more fluids to try to replenish that which is being lost in the forming urine. There are other types of symptoms that are commonly also seen with diabetes, particularly untreated diabetes. Weight loss is a big one. As I mentioned before, the person literally begins to waste away as the body attempts to utilize whatever energy sources it has, be they amino acids from muscles or lipids from stored fat tissue, to try to fuel the body through gluconeogenesis. We also get something called ketoacidosis. And ketoacidosis is it's a side effect of the utilization of fats in gluconeogenesis. When the body is using fats as an energy, as a potential energy source for the manufacture of sugars, a byproduct produced from this are ketones, which is a class of organic molecules. Remember, ketones have a double bonded oxygen to a carbon. And these ketones, with a carbon on either side, and these ketones begin to accumulate in the blood, and they produce an acidic condition to the blood called ketoacidosis. And you can actually, in untreated diabetics, these ketones normally have a sweet smell to them. They have a kind of a fruity, sweet smell to them. And a person an untreated diabetic that develops ketoacidosis can actually have such high levels of ketones in the blood that some of them get released or excreted in the mouth. And when you smell their breath will smell really fruity, sweet because of those ketones. And this can actually lead to another condition called Kussmaul respirations, which is basically a type of labored breathing, and it's a means by which the body is trying to compensate for the acidic environment of the blood by blowing off some carbon dioxide, which in an indirect way, which hopefully you remember from Bio 202, assists in helping to recalibrate the pH levels of the blood. And of course, we get hyperglycemia. We already talked about that in increased levels of sugar present in the blood. Now, diabetes mellitus not only impacts sugar levels, it has a lot of other deleterious or damaging effects on many organs in the body. And in another part of this lecture on diabetes, I'm going to talk about those. So I'm kind of putting a placeholder in here to remind you that later on we will talk about other types of impacts of diabetes mellitus on organs of the body. But for now, I want to go on and talk just a little bit about the pathogenesis of type 2 diabetes, which frankly is far more complicated than the pathogenesis of type 1. And there's still plenty of debate in the research community about the nuances of how type 2 diabetes develops, and which causative agent maybe is a triggering event, or is it a collection of triggering events that all have to happen within a relative time period of one another to initiate type 2 diabetes. This is all kind of still up for argument, but I'm going to kind of outline a little bit of what some of the most prevailing hypotheses are. You'll remember that type 2 diabetes, or adult onset diabetes, accounts for roughly 95% of all cases of diabetes, and it's principally associated with obesity. In fact, obesity is probably the #1 risk factor for developing type 2 diabetes. And as I already said, this is we think it's a pretty complex interaction among a number of different triggering events that together conspire to develop type 2 diabetes. Now remember, type 2 diabetes is a type of diabetes wherein the individual initially makes perfectly normal levels of insulin. That's not the problem. The problem is that the target cells and target organs, for one or another reason, are not responding appropriately to the insulin that's being manufactured. So even though we've got plenty of insulin present, we've got sugar in the blood, we're just not getting a response to that insulin that allows for the decrease of blood sugars in the blood. So let's take a look at some of the possible triggering events that might cause this. Central obesity is the biggie. Okay, more than 55% of the population in the United States, more than 55%, are obese. And obesity is one of the primary principal risk factors for the development of type 2 diabetes, which should tell you something very scary about where our population in the United States is heading down the road. Many have already developed type 2, many more will develop type 2. Why central obesity is implicated in the development of type 2 diabetes is still debated, but there are a number of factors we think that may contribute to this, including the fact that we know the adipocytes, or fat cells, produce a chemical called, or a protein, I should say, or a hormone called resistin. Resistin, as the name implies, it influences the body's ability to respond to insulin, and it actually causes cells to resist the effects of insulin, to more or less ignore the effects of insulin and thus not take up sugars. We also think that adipocytes are responsible, and this should make sense, the more fat cells you have, and the more fat that's inside each of those fat cells, the more likely you're going to have excess free fatty acids being released into the blood. And this contributes to the formation of type 2 diabetes sequelae, or follow-ups to the problem. And the principal follow-up problem that the excess free fatty acids cause is shown right here. The more free fatty acids I have in the blood, the more I'm going to increase the levels of bad cholesterol, or LDL cholesterol, in the blood. The more my HDL, or good cholesterols, are going to decline. I'm going to get the production of atherosclerotic plaques in blood vessels, and I'm going to get a condition in the blood called dyslipidemia, which it means an imbalance of lipids in the blood. And these all contribute to cardiovascular disease, which is one of the most damaging sequelae or follow-ups to type 2 diabetes. We also think that fat tissue, the more fat tissue you have, the more you have a decrease in the amount of production of something called adipolectin. Now, adiponectin is one of a number of different hormones that are manufactured from fatty tissue. A lot of people think fatty tissue is just a place to store fats, but we've actually learned in recent decades that fat tissue does a lot more than just store lipids. It's actually a pretty active endocrine organ in and of itself. And it manufactures a number of different what are called adipocytokines. Adipocytokines. And these include things like resistin, which I already mentioned, and they include another molecule I'm going to talk about right now called adipolectin. Adiponectin is a hormone that is manufactured by fat tissue, and it helps to sensitize cells to insulin. What's weird about this is the more fat tissue you have, the less adiponectin produce. Now, if you stop and think about this for a second, it actually makes sense. Let's imagine you're a person that has very little fat tissue. Your adiponectin levels are going to sensitize cells to insulin. You're going to take up sugar. You're going to store sugar in cells, including storing sugar in fat cells. Okay? As your fat cells get more and more get larger and larger with greater and greater amounts of fat stored within them, they begin producing less and less adiponectin because they don't want to have the ability to be sensitized to insulin. If they were sensitized to insulin, they'd take on more sugar and convert it to more fat. Okay, so it's basically the body's way of saying, look, our fat cells are already full. We don't need to take in any more sugar and convert it into fat, so we're going to reduce the amount of adiponectin we're manufacturing. The problem is adiponectin not only serves as an autocrine to actually self-regulate those fat cells secreting the adiponectin, but it gets released generally into the bloodstream, and so not only does it induce insulin desensitization for the fat cells, it also includes it also makes the other cells in the body less sensitive to insulin, and that contributes to the problem. Both of the effects of resistin and of decreased levels of adiponectin collectively contribute to this idea of insulin resistance, which is the key factor of type 2 diabetes, as I said before. It's an inability to respond appropriately to levels of insulin. Now, some people have said that also perhaps genetics plays a role in this, because we do know people that come from families with diabetes in the family are more susceptible or more likely to develop diabetes themselves. And there's been some suggestions that this may have to do with genetic abnormalities in insulin signaling pathways. But the bottom line is the research that's been done on this shows that those pathway issues are not sufficient in and of themselves to cause a development of type 2. You absolutely have to have increased insulin resistance due to obesity to finally get the trigger that pushes somebody over the edge. So really, obesity is the principal predictor for type 2 diabetes development. And it should be noted, I should note here, that the type of obesity most dangerous is what's called visceral fat. Okay, the type of obesity that involves visceral fat is when people have kind of an apple shape to them, I call it. It means that they carry most of their fat tissue around their abdominal area in their viscera. People that have, or women, for example, that have what people call saddlebags, where they carry the majority of their fat in their hips and legs, are at much less risk of developing type 2 diabetes. And we think that's because visceral fat more actively secretes these resistant compounds. In other words, the visceral fat is a more active endocrine-like organ than is the fat that's associated or found in places like the hips and thighs. So what are the types of symptoms that result from untreated diabetes? Well, I've mentioned a lot of these already, so I should be able to go through this pretty quickly. In type 2 diabetes, and that's what we're talking about here principally, these are often nonspecific. And I'll give a great example of this. My stepfather developed type 2 diabetes in the early 90s, and it took a long time for us and for his physician to figure it out, because the types of symptoms he had they just didn't point to diabetes right away. He was a little overweight. He was very cranky all the time, which is another common characteristic. And some people put it off as being, oh, I'm just tired, so I'm cranky. Well, they are tired because they don't have access to sugar stores. But they also oftentimes will have these kind of almost depression-like symptoms or irritability, which I forgot to put on here. And because many of these symptoms I just talked about are nonspecific, they often get put off as something else. Oh, you're fatigued because maybe you're a little anemic, or maybe you're a little irritable. You just didn't get enough sleep last night, or something like that. They're often nonspecific. Some of the other characteristics are dyslipidemia. That's where we have an imbalance in the levels or ratios of, for example, HDL, the good kind of cholesterol, versus LDL, the not good kind, and the presence of triglycerides at abnormal levels in the blood also are part of dyslipidemia. Hyperinsulinemia, overly high levels of insulin in the blood. Remember, if in type 2 you're not responding to insulin, the pancreas will continue to produce insulin in hopes that now the cells will respond to insulin, which they're not. Hypertension can develop. Part of this has to do with the fact that if I have more glucose present in the blood, glucose increases the osmolality of the blood, which means it draws more water to the blood. Blood volume goes up, and as blood volume goes up, blood pressure goes up. Also, these people sometimes are susceptible to recurrent infection, in part because, if you think about it, you have very high levels of sugar in the blood. Bacteria thrive on glucose. So if you get any kind of damage to tissue and there's release of blood into that area where the tissue damage occurs, well, you get a flooding of the area with glucose, which can fuel bacterial invasion pretty efficiently. Also, people with type 2 diabetes and type 1, by the way, can often develop what are called paresthesias. There should be one more. Oh, I did put it in there, paresthesias, 2 S's there. Paresthesias basically mean any kind of like a tingling sensation. They say they have like numbness in their feet in particular, but sometimes they'll have it in their hands too. This is due to a condition called diabetic neuropathy I'm going to talk about in a few minutes. It has to do with damage that's occurring to neurons. So I want to talk very quickly about the types of ways that we can treat type 1 and type 2 diabetes, and we're going to stop it after this slide and pick up again on a third part of diabetes mellitus here. You all know that for type 1 diabetes, that's where people cannot manufacture insulin, that the number one means by which it's treated is through insulin injection. But how many of you have ever thought, why is it that people with type 1 diabetes have to inject their insulin? Why can't they just take a pill? If you've ever wondered about that, well, here's the answer. Insulin is a hormone that happens to be a peptide hormone. That means it's made of amino acids. What happens to proteins, to peptides in your food when you consume them? Well, of course, they get broken down somewhat by pepsinogen in your, or pepsin in your stomach. Then they travel onto your small intestine, and we had all those different pancreatic enzymes and brush border enzymes you learned about in 202. Things like trypsin and chymotrypsin and carboxypeptidase and aminopeptidase and dipeptidase that all conspire to degrade those peptides down into individual amino acids. So if you gave a person an insulin pill, it would just get digested. That's a protein. It would just be digested into individual amino acids and absorbed into the body and would have no impact or help or benefit to that type 1 diabetic. So they have to inject insulin directly. That's the only way that they can absorb it as the whole insulin molecule without having it destroyed. Now compare that, for example, with birth control pills. Birth control pills contain principally estrogen and progesterone. Estrogen and progesterone are both based off the molecule cholesterol. Cholesterol happens to be a molecule that can be absorbed directly when consumed. It does not get broken down. Lipid digestion in the body is much simpler than protein digestion is, and so you can therefore take a pill that contains progesterone and estrogen and have that estrogen and progesterone make its way into the blood unaltered during digestion. For type 2 diabetes, there's a lot of different types of drugs that are utilized for type 2 diabetes to treat it. Some of them try to increase the response of cells to insulin. Others try to increase the amount of insulin that's being released. There's a variety of different mechanisms, but only in the more advanced cases of type 2 diabetes do people start having to take insulin injections. Oftentimes they can modulate type 2 diabetes or control it through a variety of different drugs. Diet modification is a big thing, right? We all know that diabetics are told not to consume lots of simple sugars because it too rapidly elevates the levels of glucose in the blood. It makes it very difficult to control the hyperglycemia. Exercise turns out to be an absolutely pinnacle, critical, important event in the control of diabetes. Here's why. And this is, I think, really interesting. Your book does not talk about it, but it's a really important point to know. When we look at cells in the body, they have the ability to take in sugar based on their response to insulin, right? So let's take just a typical cell here. Okay, so here's a typical body cell. When insulin, here's my insulin, when insulin binds to, here we show it binding, to its insulin receptor on the surface of the cell, a series of events happens in a typical body cell. And these involve getting some secretory vesicles that contain a certain type of protein called a GLUT4 protein. GLUT stands for glucose transporter protein, to migrate to the surface of the plasma membrane and to integrate then those GLUT4 transporter proteins into the surface of the plasma membrane. And once those GLUT4s are integrated into the plasma membrane, they can begin shuttling glucose molecules into the cell. Now, most cells in the body, somatic cells, have GLUT4 transmembrane or transporter proteins. That means that those GLUT4 proteins are normally housed in vesicles, and it's only when insulin is bound to the insulin receptor on the surface of the cell that those GLUT4s migrate to the surface of the cell and begin taking in glucose. Once insulin levels diminish, those GLUT4 transporter proteins get taken back into the cell and repackaged inside a vesicle until the next time insulin is secreted. Now, why am I telling you this? Well, I'm telling you this because exercise has a very interesting impact on this particular mechanism. It turns out that in skeletal muscle, the simple act of contraction of those muscles is sufficient to induce the installation of GLUT4 proteins on the surface of the cell without first having insulin give them permission to get to the surface of the cell. So in essence, what exercise does is it allows skeletal muscle to take in sugar without first waiting for permission from insulin to do so. This is a big reason why exercise is so incredibly beneficial for diabetics. Exercise is also beneficial for type 2 diabetics for another reason. The more you exercise, the more you decrease your adipose stores, the amount of fat tissue you have. And if you decrease the amount of fat tissue you have sufficiently, you can actually cure type 2 diabetes. You can actually get a person to go from being a type 2 diabetic to normal blood sugar levels again if they can get rid of sufficient amounts of fat. One other thing I want to mention about GLUT4 transporter proteins. GLUTs, there's more than just type 4. There are actually, at this point that we know of and understand, at least 6 different types of GLUT proteins. There's GLUT1, there's GLUT2, GLUT3, GLUT4, etc. And it's interesting to note that some different types of GLUTs are, for example, always present on the plasma membrane. They're not stored inside vesicles and then put on the plasma membrane at certain times. For example, in the brain has what are called GLUT1 and GLUT2 proteins, and those are more or less always installed on the surface of neurons so that those neurons have constant access to glucose regardless of what's happening with insulin levels. So different types of organs in the body have different types of GLUT transport proteins, and they respond differently to insulin, or they may be able to take up sugar without responding to insulin at all. It just depends on the type of organ. Okay, that's good enough for now. Let's continue this discussion on the next lecture.